Anybody have any questions? It kind of creeped up on me. I didn't realize we've got another second exam coming up a week from Friday. So it's going to be over enzymes, energy, and cell respiration.
We think we'll finish all the cell respiration.
Yeah, we will finish it. Okay, just a couple of words to finish up with enzymes. We were talking about this phenomenon at the very end of the period, which provides an opportunity to regulate metabolism and for the cell to have some control over how it uses its various resources, here represented by some compound designated L. L is kind of a versatile compound in cellular metabolism in this example, because it can be converted into a number of different end products. And sometimes the cell might need this one, sometimes this one, sometimes this one. One of the instances where it probably wouldn't be advisable to waste L to make S, for example, is if the cell already had a sufficient amount of S. Okay? So there's this phenomena that can take place called feedback inhibition, this self-correcting process that ensures that if you have too much of this end product, that can go back and shut off an enzyme that is responsible for catalyzing the pathway that produces that product. So it's much like the example I used with the heater. If it gets too hot, the heater turns off. Okay? If too much S builds up, then S can interact with an enzyme that is responsible for producing S. Let's give you a particular example using a particular metabolite. Threonine, anybody know what threonine is? It's an amino acid. Okay, it's one of the 20 amino acids. Anybody know what isoleucine is? It's another one of the 20 amino acids. Okay? Cells metabolically have the ability to convert threonine into isoleucine. So if you eat protein that contains this amino acid, if you need it, your cells can convert this one into this one by this complex metabolic sequence. It involves several intermediate steps, each of which is regulated by an enzyme. The enzyme we want to concentrate on is the very first enzyme in this sequence, threonine deaminase. Okay? So, if the cell doesn't have much isoleucine present, and it needs all of the 20 amino acids to build proteins, so when you've got those ribosomes reading a message, if there isn't this critical amino acid around, it can't usually build proteins because most proteins will contain this amino acid. So it's important that the cell has this. So, threonine deaminase grabs some threonine, binds at the active site, converts it into this, another enzyme to this, etc., and the end product isoleucine is produced. Okay? And if this is constantly being used up in protein synthesis, then this sequence is just going to continue. But what happens if we ingest some protein and it's got a lot of isoleucine in it? Then do we want to waste this resource making something we already have? No. Okay? What can happen is this enzyme right here, the first enzyme in the sequence, has a separate site from the active site. What do we call that separate site? Allosteric. It's the other site. Allo means other. And isoleucine, the end product of this metabolic sequence, can bind to this active site, this allosteric site, sorry. And when it does, that changes the shape of the active site, so threonine can't bind. So the presence of the end product, in this particular case, inhibits the synthesis of that particular end product. What happens if the cell starts to use up all its isoleucine? Then there's none to bind to this allosteric site. And if the allosteric site is not occupied, then the active site reverts back to its normal shape, and it's able to bind threonine, and it will make more isoleucine. So do you see how the cell can regulate how much of this it produces by this feedback inhibition mechanism. Okay? As it turns out, feedback inhibition is not the only type of regulation that can take place. There is also a phenomena where molecules that are end products of pathways can be activators of enzymes and actually speed up those enzymes. So there's both feedback inhibition and activation. And we'll come back and talk about a concrete example of that in a couple of days as we dig into cellular respiration. So that pretty well wraps up what we need to cover about enzymes for now. So let's shift gears and head over to cellular respiration. The reading quiz Monday will be over the first three sections of Chapter 7, 7.1, 7.2, and 7.3. And that will take us through glycolysis and the citric acid cycle. It will not include the section on ATP synthesis by chemiosmosis. Okay? I'll get you the page numbers tomorrow, so you've got those page numbers, but 7.1, 2, and 3. Okay, so we've talked about the fact for the last couple of days that energy is needed constantly to maintain the low state of entropy of a cell, for a cell to maintain its organization. And without ATP to do that necessary work to maintain that low state of entropy, cells become disorganized, and if a cell becomes disorganized, it's dead. We distinguished also between how cells could obtain forms of chemical energy that could be used to produce ATP. If you make that chemical energy specifically in the form of glucose molecules by the process of photosynthesis, then you're said to be an autotrophic organism. You're a self-feeder. You produce your own glucose. Okay? If you're like us and you can't produce glucose because you can't do photosynthesis, then you are a heterotroph. You have to feed on other organisms and obtain organic molecules from them, and then you can use those organic molecules by cell respiration to make ATP. So we're going to concentrate on the phenomena now of cell respiration and see how organic molecules, and we're going to concentrate on the organic molecule glucose, can be chemically converted into a usable form of energy that the cell needs to do cellular work. And that is the molecule ATP. Okay? So here's our balanced reaction for aerobic cellular respiration. Aerobic cellular respiration indicates that oxygen is present. We're going to see later on that there are some other types of cellular metabolism that produce ATP that don't involve oxygen. You studied one of them last week in lab when you did experiments with fermentation. So we're going to distinguish between fermentation and aerobic cellular respiration. But here is the equation for aerobic cellular respiration. So we've got this molecule glucose. And what's significant about glucose? It's got a lot of hydrogen. What does hydrogen represent? Electrons, potential energy. Okay? In the presence of oxygen, those hydrogens or electrons can be ripped off of the glucose, forming carbon dioxide. And in the process, oxygen becomes converted into water molecules. So basically, we're moving some hydrogens around. Okay? We're moving them from glucose to oxygen. The resulting products are carbon dioxide and water. The free energy change of this, or the delta G, is minus 140 kilocalories per mole. So what does that indicate? Is this an exergonic process or an endergonic process? Exergonic, right? So the free energy of glucose is up here. The free energy of carbon dioxide and water is down here. So this is a downhill process, spontaneous or not? Spontaneous, okay? But in the absence of enzymes, the rate of this reaction is really, really slow. That'll be important. What forms of energy are liberated as this energy conversion takes place? Well, ATP is produced, but a fair amount of heat is produced too. Is the cell going to be able to use that heat to do meaningful work? Nope, that's considered a waste product. I've seen some calculations, and of all of the energy that's present in the glucose molecule, somewhere around, I believe, around 30% is converted into ATP. So it's not a particularly efficient process, but we get what we can get out of it. Okay? ATP is produced by the phosphorylation of ADP. This particular reaction is endergonic, so it's going to have a positive delta G. So this particular endergonic synthesis of ATP is going to have to be coupled to this exergonic breakdown of glucose. Remember that if reactions are paired together, then that the overall delta G of the coupled reactions is the sum of the individual reactions. So glucose is producing enough energy that can drive the synthesis of ATP molecules. So it's a pretty good system. One of the things that you should notice about this particular reaction is that some molecules are losing hydrogens and therefore electrons. So what's happening to glucose? Lose electrons. Leo. Oxidized, right? So glucose is becoming oxidized, and the resulting product of that oxidation is CO2. What's becoming reduced? Oxygen is picking up those electrons in the form of hydrogens and forming water molecules. So glucose is cellular respiration is an oxidation reduction reaction. Now, one of the things that we're going to see is that what really gets oxidized and reduced during the reactions of cellular respiration are some important coenzymes. Remember that coenzymes are partners that work with enzymes. And these particular molecules have the ability to pick up hydrogens and then get rid of them, to become reduced, to become oxidized. And that's going to be really important to understand as we dig through some of the details of cellular respiration. Okay, well, what we want to do is we want to try to harvest some of this energy that is released during the exergonic breakdown of glucose. Some of that 140 kilocalories per mole of energy. There's really two ways that we can store the energy from the breakdown of glucose. And one of the ways is by reducing this molecule, NAD+. Remember that we said when something becomes oxidized, it loses energy, and when a molecule becomes reduced, it gains energy. Okay? So the reduction of this NAD, sticking some electrons onto it, or hydrogens, is a way to store some of that energy. The other way to store some energy is to phosphorylate ADP into ATP. So those are going to be two ways that we're going to have to understand that we harvest some of the energy from the breakdown of glucose molecules. Okay, so we've already talked about oxidation reduction, so we don't have to go through this again. So an oxidation reduction reaction, some kind of a compound loses electrons or hydrogens and becomes oxidized. And another compound then will pick up those electrons or hydrogens and become reduced. So in the case of cellular respiration, glucose becomes oxidized into carbon dioxide. Oxygen becomes reduced into water molecules. These reactions always occur in pairs. oxidation reduction in cellular respiration typically involves coenzyme molecules. And there are two coenzymes that we're going to be interested in cellular respiration. One is called NAD, and then its kind of cousin is FAD. If you understand NAD, then FAD is pretty similar. The important thing to understand about these coenzymes is that they can exist in one of two states. NAD can exist in the oxidized state or the reduced state. So it can go back and forth between the oxidized and reduced state depending upon whether there are electrons added to this molecule or whether they're taken away. I know this looks a little bit confusing right now. Why is this written as NADH plus H plus? Sometimes you'll actually see that written, I'll do this a lot, as just NADH2. I'll explain what the difference is between these two representations of reduced NAD. Sometimes you'll actually see it written this way too. All of these three representations represent reduced NAD. This is actually the most accurate of those representations. Another critical thing about coenzymes to understand is that they're present in the cell in very small amounts. Many of the coenzymes in the cell are produced by metabolic pathways. The cell synthesizes them. And the starting material is obtained from vitamins. So there's a B vitamin, for example, that is used metabolically in the cell to make cellular NAD. You don't need many vitamins, do you, to be healthy? You need those in tiny amounts. That's because these coenzymes are present in the cell in limited or small amounts. So keep that in mind. There are some enzymes in the cell, particularly a group of enzymes called dehydrogenases. Anytime you see this prefix D, it means without. So dehydrogenases are an enzyme that is going to catalyze a reaction that removes a hydrogen from some substrate. And in the process, oxidizes it. Okay? So we're going to see, for example, that there in glycolysis, there's this enzyme we'll talk about called pyruvate dehydrogenase. That is an enzyme that takes some hydrogens off of a substance called pyruvate. Well, a lot of these dehydrogenases have to be paired with a coenzyme. And the coenzyme that they have to be paired with is the oxidized form of the coenzyme. So where does this hydrogen go when it's ripped off of X? It's picked up by the oxidized coenzyme, and the oxidized coenzyme then becomes reduced. Okay? Well, if there's a limited amount of this in the cell, we quickly reduce all of the NAD. So we run out of this. And if we don't have this present, this enzyme doesn't work. So this reaction would then stop. So one of the things that's going to be really important in understanding cellular respiration is the understanding that the cell has got to have some way to keep a good supply of this to keep many of the reactions of cellular respiration going. How can it do it? It doesn't just get more NAD. You don't have to ingest more NAD or make more. What can you do? You can recycle the NAD from its reduced state back to its oxidized state, but then those hydrogens have to go somewhere else, right? Because if you take them off of this molecule to reform this, so this reaction can keep going, where do those hydrogens go? from the reaction of cellular respiration, where do those hydrogens go? They're ultimately going to be picked up by oxygen, and water's going to be formed. So the cell, to keep respiration going, doesn't need a constant input of NAD. It can recycle if it has a constant input of what? Oxygen. And if you don't have oxygen, then you can't recycle. There's no place to dump these hydrogens off to. And therefore, you run out of this, because it's all in this form. And therefore, this reaction, or those reactions, don't occur. And cellular respiration stops. And if cellular respiration stops, what can't you produce? Can't make ATP. And what happens to cells? They die. Okay? So this is why oxygen is so important in cellular respiration. It's the final resting place, if you like, for the electrons that were stripped off of the glucose molecule during cellular respiration. And as those electrons are moving from one molecule to another, energy is produced. And specifically, energy is being produced in large amounts in the form of ATP. If this is confusing you still a little bit, we're going to go back through this several times. We will keep hammering away at this. So these are the things that you need to know right now about coenzymes. Types, the fact that they can become oxidized or reduced depending upon the situation. The fact that the oxidized form is critical to keep cellular respiration going. Okay, here's what NAD looks like. Now, I'm not going to make you draw this. I just want you to see what the molecule looks like. NAD stands for nicotinamide adenine dinucleotide. Look familiar at all? What does it kind of look like? Nicotinamide adenine dinucleotide. Adenine. A, as in the A, T, G's, and C's. Okay? This is adenine. This is a sugar. This is a phosphate. Kind of looks like a nucleotide, doesn't it? Okay? The part of the NAD molecule that we're most interested in is this nicotinamide portion up here. Because this is where the oxidation and reduction is going to take place. This is where hydrogens are going to be removed or added. This is the oxidized form. This is the reduced form. If you add hydrogens to this molecule, if you take some hydrogens off of a food molecule like sugar, then the coenzyme NAD, which is oxidized, can pick up those hydrogens and become reduced. Now, in reality, what are hydrogens? A hydrogen is just a proton and an electron. So two hydrogens are two protons and two electrons. What happens? When this becomes reduced, two electrons are added to this molecule. One proton is added to it, but there's one proton left over. So the reduction of NAD involves the addition of two electrons, one proton, and there's a leftover proton that isn't being used. So that's why it is best to represent this as NADH. Okay, here's one electron, one proton. Another electron is added to this nitrogen over here, turning it from a plus to a no charge. NADH plus H plus. If you prefer to represent reduced NAD in this form or this form, that's fine with me. I just wanted to show you this because it's going to come up. You're going to see it written more than one way in either PowerPoints or notes or textbooks. If we oxidize this reduced form of NAD, you're going to have to pull off a couple of electrons and a proton, and they're going to have to go someplace. So NAD, oxidized form, reduced form. FAD is really similar. The only difference is that FAD actually accepts two hydrogens. So reduced FAD really is represented correctly as FADH2. It picks up two hydrogens. So here's some molecule containing hydrogens. Here's a dehydrogenase enzyme that's going to rip one of those hydrogens off of that molecule. Here's the coenzyme in the oxidized state that the dehydrogenase requires to function. If this isn't present, this doesn't work. Hydrogen's removed, the substrate becomes oxidized, and in the process, the NAD coenzyme picks up protons and electrons, becomes reduced, and there's this extra proton hanging out there that's not used in the process. Okay, we said there were two ways that you could harvest energy released from cellular respiration. The other way is by making ATP. ATP synthesis is endergonic. It's uphill. It involves phosphorylation of ADP. So for this to work, to phosphorylate ADP, we're going to need some source of phosphate. And we're going to need some input of energy, because this is an endergonic reaction. There's two different mechanisms that the cell uses to synthesize ATP during cellular respiration. One is called substrate level phosphorylation, and the other is called oxidative phosphorylation. So let's distinguish between those two. Here are some important things about substrate level phosphorylation. Substrate level phosphorylation involves an enzyme catalyzed reaction. And this enzyme basically is going to grab a phosphate group off of one molecule and stick it onto another one. Specifically, it's going to stick it onto ADP. So, here's an enzyme. Here's some kind of substrate that has a phosphate group attached to it. This enzyme has a binding site for both the substrate and ADP. And the enzyme catalyzes a reaction where that phosphate group is removed from this and added onto this to make ATP. The substrate is converted into some new product then because it doesn't have a phosphate group anymore. So this is an enzyme catalyzed reaction, and the molecule that the phosphate group is taken off of is some organic molecule. This might not seem relevant right now, but it will be. This particular reaction does not require any membranes to occur. You can get this to work in a test tube if you have a phosphorylated substrate and the proper enzyme and ADP. A pretty significant thing about this. Substrate level phosphorylation in a cell produces a fairly small amount of ATP. This is a number that you're going to have to be familiar with. This 38 represents the maximum number of ATP that can be produced by a molecule of glucose during aerobic cellular respiration. And there's actually going to be some accounting that we're going to go through to add up to see how you can get to this 38 total. Substrate level phosphorylation produces only a total of 4 of those 38. Okay? So a pretty small percentage. So an enzyme catalyzed reaction that doesn't require membranes that produces a fairly small amount of ATP. Oxidative phosphorylation. This one's a little more complicated, and this one's going to require a little work on our part to wrap your head around. This particular process still has energy requirement. This synthesis of ATP is still anorganic. But what drives that is the energy stored up in a hydrogen ion gradient. When we have a higher hydrogen ion gradient, we have a high level of hydrogen ions in one place, and we have a low level of hydrogen ions someplace else. This represents potential energy. We have a membrane separating two spaces or two compartments. I've introduced that concept when we talked about mitochondria. This is literally like that slide that I showed when we started the discussion of energy, and particularly the difference between potential and kinetic energy. This is like the water behind a dam. This is your reservoir. This is your dam. This is the area below the reservoir. What's the natural tendency of matter going to be? How are these hydrogen ions going to want to behave? Where do they want to go? To their low concentration or their low state of free energy, right? So this is a state of high free energy. This is a state of low free energy. But we got this membrane here. Okay, think about permeability properties of membranes. Are membranes going to be very permeable to hydrogen ions? Nope, they're small. But they're what? They're charged. And those charged protons are just not going to want to go across that phospholipid bilayer. So that membrane is pretty impermeable to these H pluses. And yet they are going to move across. Because inserted into that membrane is a channel protein that allows those protons to flow across. And this channel protein is also an enzyme. It's an ATPase enzyme. The enzyme that catalyzes this reaction is ATPase. So this is almost like that spillway, that gate in the hydroelectric dam. The water can flow through, and when it flows through, it spins the turbine that generates electricity. When protons go through this ATPase channel, basically the energy is released to drive this synthesis of ATP. So you need a membrane for this oxidative phosphorylation to occur. Yeah, you've got to have something that separates the high from the low concentration and helps to maintain that gradient. The phosphate that's going to be transferred in this particular case is an inorganic phosphate. It's not attached to some organic molecule. So there's that subtle difference. This particular process of oxidative phosphorylation is where we get our real payoff of ATP during aerobic cellular respiration. Out of the 38 total, 34 come from this process. Remember, only 4 net came from substrate level phosphorylation. So this is where you get the big payoff. Now, what we're going to spend some time talking about is how do we build up this water behind the dam? How do we build up this hydrogen ion gradient? Well, as it turns out, that's going to be coupled to this oxidation reduction of NAD. So those are going to be tied together. We'll tell you how that works later on. So, here's our membrane. Here's the space inside the membrane. Here's the space outside the membrane. In oxidative phosphorylation, we build up a high concentration of hydrogen ions here, and there's a low concentration of hydrogen ions here. Here's our channel protein, our ATPase enzyme. So as those protons or hydrogen ions flow through the channel protein from high concentration to low concentration, that's coupled to the phosphorylation of ADP to make ATP. Don't worry about this particular part right now. This is representing how we build up this hydrogen ion concentration right here. And it involves hydrogens moving from one coenzyme to other molecules. Now, when I first gave you the equation for cellular respiration, the balanced equation for cellular respiration, it just said glucose plus oxygen to CO2 and water. And it implied that it was just a one-step reaction. In reality, there are multiple sequences within cellular respiration. So that was the overall equation. And we're actually going to see a fair number of the individual steps. But it's important to get some kind of an idea of why cellular respiration is organized into these sequences of individual steps as opposed to just one step. Here's an example. Okay? I can combine hydrogen and oxygen under certain conditions to form water. And if you do this as a one-step reaction, hydrogen and oxygen have a higher free energy than water. And as these are combined to form water, we get all of that energy released in basically one step. There's kind of this explosive release of heat and energy. The same thing basically happens in cellular respiration. We've got hydrogens from food that are eventually combined with oxygen to make water. But the energy is released in small increments as these steps occur one by one by one. It's hard to harness energy from this single burst of release all at once. In cellular respiration, there are several conversions that take place that release little bits of energy at a time in the form of ATP, which the cell can then harness and use to do work. Let me give you kind of a silly example. The goal of your car is to take you someplace, right? And the car is powered by a fuel, a hydrocarbon. And in the process of burning that fuel, the car moves. Well, one of the ways you could get your car to move would be to go out to your car, open up the gas tank, and throw a match in there. And that fuel would all be converted into energy in one explosion. And the car would move, but then it would be done. Okay, it'd be destroyed. Instead, what happens in a car? Well, there are little mists of fuel injected into cylinders in your engine where controlled burnings take place, and you have these little kind of mini explosions which drive pistons, which turns a crankshaft, which moves your car without destroying it. Those tiny, small bursts of released energy that can be harnessed and utilized safely, as opposed to just the one big explosive release. So cellular respiration has kind of a step-by-step conversion of glucose into carbon dioxide and water, and along the way, small amounts of energy are released, which can be harvested safely to do cellular work. I mean, there are over 30 individual steps in cellular respiration, depending upon how you count things. To make sense of this all, we're going to break cellular respiration down into 3 general types of reactions, or 3 phases. And those particular phases are called glycolysis, citric acid cycle, also sometimes called the Krebs cycle. Hans Krebs is the guy who figured out how this set of reactions works. And then the electron transport system, or sometimes also called the electron transport chain. One of the things that I'm going to urge is that you not get overly consumed by the details in this information and lose sight of the big picture. Okay? That's really a risk sometimes when you get into metabolism, is you get so hung up on all of the details, you don't really understand big picture what's going on. Okay, you lose the forest for all the trees. So as we go through this, I'm going to show you some chemical structures, but I'm not going to make you memorize them. Okay? But I think it's important to see the rearrangements that are taking place if you can kind of begin to understand what's going on overall in this particular process. So what I'm going to do is I'm going to kind of break things down for you. And this might look like kind of a long list. But in reality, it's not that bad. And this really is a very good way to study for a lot of this stuff about cellular respiration. So we're going to concentrate on these 3 reactions: glycolysis, Krebs cycle or citric acid cycle, and the electron transport system. What do you need to know? You need to know what each of these begins with. What's the starting material? You need to know what it ends up with. The detail in between, don't get too hung up on. Okay, so we're going to look at glycolysis, for example, and we'll see the sequence of conversions that takes place. But starting product, end product. Is carbon dioxide produced? Okay? Because in cellular respiration, glucose is completely oxidized into carbon dioxide and water. But not all of these sequences produce CO2. So does glycolysis produce CO2? Does citric acid cycle produce it? Is there reduction of NAD involved in the sequence? Is there oxidation of NAD involved? Same for FAD. Where do these take place? These take place in different cellular locations. If ATP is produced, how's it produced? By substrate level phosphorylation or oxidative phosphorylation? And if it's produced, how much? So you have to be able to do a little bit of accounting. Is O2 directly involved in the process? And is there any ATP used during the process? Okay? So these are the types of questions that you should be able to answer about glycolysis, citric acid or Krebs cycle, and the electron transport system. And we're going to be hammering away at these as we go through these particular processes. So here's what it looks like overall. This is kind of the big picture of cellular respiration. We're going to start out with glucose. And during the process of glycolysis, glucose is going to be converted into something else called pyruvate. That particular set of reactions produces some ATP. And it's all by that process of substrate level phosphorylation. So are the numbers here going to be very big? No. Substrate level phosphorylation doesn't produce much ATP. This is also involving an oxidation of glucose, so some hydrogens are going to be removed. They're going to be picked up by the coenzyme NAD. So there's going to be some reduction of NAD. If oxygen is present, the pyruvate that's produced by glycolysis will enter the mitochondria. These reactions are happening in the cytoplasm. This process is happening in the part of the mitochondria called the matrix. What happens in the matrix? Pyruvate is completely broken down into carbon dioxide and water. And some more ATP is produced by substrate level phosphorylation. So is it going to be very much? No, a little bit more here. Pyruvate has hydrogens, carbon dioxide doesn't. So those hydrogens that are removed are going to be picked up by NAD and its cousin FAD. There's no FAD here. So this is a new coenzyme. Okay? So we're going to get some more electron carriers reduced. So what's going to quickly happen as glycolysis and citric acid cycle occur? The oxidized form of NAD is going to get converted into the reduced form. And we're going to run out. Without the oxidized forms, this set of reactions can't occur, this set of reactions can't occur, it stops. And then we don't even make this little bit of ATP. Fortunately, in aerobic cellular respiration, there's a third set of reactions, the electron transport chain. And this takes place in the mitochondria as well, but this takes place associated with the inner membrane of the mitochondria. This guy right here. And during the electron transport chain, the hydrogens are stripped off of these coenzymes, and where do they go? They go to oxygen to make water. And when you strip the hydrogens off of NAD and FAD, what do? You oxidize them. So now those coenzymes are available to participate in these reactions, so these can keep going. As we do this process of electron transport, and we recycle NAD from its reduced form to its oxidized state, we build up a hydrogen ion gradient. We'll tell you how later. And that hydrogen ion gradient is used to make ATP by oxidative phosphorylation. And this is where we get the big payoff. This is where we get most of our ATP. Okay? We will continue here on Friday.